Imagine watching two people uh, run an identical, highly complex obstacle course.
Okay.
Like, one is a perfectly healthy athlete, and the other is, nursing a severe knee injury.
Right. So very different physical states.
Exactly. Now, if you wanted to compare their performance, how would you accurately map their progress side by side?
I mean, it sounds straightforward at first.
Right. You just compare their times of each obstacle.
Mm.
But what happens when the injured runner approaches a massive wall climb, realizes they physically cannot do it, and just completely skips that obstacle to move to the next one?
Oh, yeah. That throws everything off.
Completely.
Yeah.
If you are tracking their data, how do you map that skipped event without completely skewing the rest of the timeline?
Well, computationally, it becomes an absolute nightmare.
Yeah.
Because if you just, blindly align their chronological checkpoints, you end up comparing the healthy runner scaling a 12-foot wall to the injured runner crawling under a mud net.
Right, because it was technically their next step.
Exactly. You are forcing a comparison between two entirely different physical realities just because they happened at the same sequential step.
And this isn't just a fun thought experiment. When a diseased cell skips a developmental step, old algorithms skew the whole timeline trying to map it.
They really do.
This exact problem is the core challenge in one of those vital fields of modern biology, which is mapping the dynamic timelines of single-cell development.
A massive hurdle.
So today, we're doing a deep dive into a computational framework that finally solves this. We're pulling from a recent Nature Methods article detailing a new tool called Genes2Genes, or G2G for short.
It's really groundbreaking stuff.
It is, and for you listening, whether you're analyzing complex data in your own field or just interested in the future of medicine, the stakes here are profound.
Yeah, understanding how to accurately map these cellular timelines is basically the blueprint for optimizing lab-grown organs.
Right, and intercepting deadly diseases before they even physically manifest.
Absolutely. But to understand the magnitude of what Genes2Genes actually accomplishes, we have to look at the computational task it's replacing.
Which is called pseudo-time trajectory inference, right?
Yes, pseudo-time trajectory inference. So when scientists use single-cell RNA sequencing, they are essentially taking a snapshot.
Like a literal photograph.
Sort of. It's a snapshot of thousands of genes inside a single cell at one frozen moment in time.
Okay, got it.
But biological processes, like a stem cell maturing into a functional lung cell, they are dynamic. They happen over time.
Right, it's not just a single picture, it's a whole movie.
Exactly. So algorithms analyze the transcriptomic similarity across thousands of these individual snapshots.
They're looking for patterns.
Right. They look at the gene expression profiles and computationally arrange them into a chronological timeline.
Like putting the frames of a movie in order.
That's a great way to think about it. And that generated timeline, that movie, is the pseudo-time trajectory.
Okay, so the algorithm takes a pile of scattered biological snapshots and infers the chronological order based on how the gene expression, subtly shifts from one cell to the next.
Precisely.
But the problem arises when you want to compare two different timelines, say a healthy cell's development versus a diseased cell's development.
Yes. And for years, the default method for aligning these timelines has been an algorithm called dynamic time warping.
DPW.
Right, DTW. Now, dynamic time warping attempts to find the optimal mapping between two time series by minimizing what we call Euclidean distance.
Okay, let's unpack that. Euclidean distance.
In high-dimensional gene expression space, Euclidean distance is essentially the straight-line mathematical distance between two data points.
So it's just looking for the shortest path.
Exactly. DTW stretches or compresses the timelines to find the closest possible match between the gene expression of the reference cell and the query cell.
Okay.
It searches for the path of least resistance to draw a continuous line connecting point A to point B.
Right. But I assume the fatal flaw there is that word continuous.
You hit the nail on the head.
DTW assumes a definitive match exists and just forces the alignment. But getting back to our injured runner analogy, what if a diseased cell completely skips a developmental stage?
Right. What if that state simply doesn't exist?
Yeah. If it doesn't exist in the query cell, wouldn't DTW just smear the surrounding data together? Like forcing a Euclidean connection that is mathematically convenient but biologically totally false.
That limitation is exactly where DTW falls apart.
Wow.
It operates on this rigid, hard-coded assumption that every single time point in the reference sequence must map to at least one time point in the query sequence.
So it literally cannot handle gaps.
It has no vocabulary for them at all. In genetics, we call these indels insertions or deletions.
Indels, okay.
If a developmental state is missing, DTW just forces a match anyway.
That seems like a massive oversight. Are there workarounds?
Well, there are post hoc processing methods. Like, there's a tool called Tragedy that tries to clean this up after the fact.
How does that work?
Tragedy runs GTW, looks at the resulting messy alignment, and then tries to snip away the worst matches based on predefined thresholds.
I mean, but snipping away bad data after the fact doesn't fix a broken foundation.
Not at all.
If the underlying math assumes a guaranteed match, trying to just threshold your way out of it when comparing data sets that have fundamentally diverged, that seems like applying a Band-Aid to a structural collapse.
It absolutely is, which is why this new Nature Methods paper introduced genus 2 genes.
Okay, before we dive into how G2G fixes this, let's just do a quick recap for you listening.
Like, good idea.
So, cells develop along these timelines called pseudo-time trajectories. Older methods, like DTW, falsely force matches between these timelines even when entire biological stages are completely missing.
Right, they just smear the data.
Exactly.
Yeah.
So my question for you, listen to this deep dive, is how often do you force comparisons in your own data or even in your life when you should really be acknowledging the gaps?
And that's a great question to keep in mind.
So how does GDG actually acknowledge those gaps?
Well, G2G entirely discards the assumption that a match must exist.
It just throws it out.
Completely.
Yeah.
Instead, it uses a dynamic programming framework that unifies dynamic time warping with biological sequence alignment.
Okay, what does that mean in practice?
Specifically, it adapts something called Goto's algorithm.
Goto's algorithm.
Right. Goto's algorithm is a staple in bioinformatics. It's traditionally used for aligning DNA and protein sequences.
Oh, okay, so it's already well tested.
Very. And its superpower is handling what we call affine gap penalties.
Affine gap penalties. That sounds intense.
It just means it understands context. It understands that opening a brand new gap, which represents a biological mutation or a massive regulatory shift, is mathematically expensive.
Expensive meaning highly unlikely or costly to the algorithm.
Oh, I see. So starting a new error is hard, but continuing one makes sense.
Precisely. By bringing Goto's logic into single-cell trajectories, G2G can handle both matches and complete mismatches simultaneously, without fragmenting the timeline into microscopic, unusable pieces.
Exactly. It keeps the big picture intact.
So it's not just stretching the timeline, it's actively evaluating whether a gap is biologically justified.
Yes.
And it does this using what the paper calls a five-state machine.
Right. The five-state machine is the core of it.
As the algorithm compares the reference timeline to the query timeline, it assigns every single moment into an alphabet of 5 distinct states: M, V, W, I, and D.
Let's define that alphabet because it's really the secret sauce here.
Let's do it.
So M stands for a one-to-one match. The cells are doing the same thing at the exact same time.
Simple enough.
Then there's V. V is one-to-many expansion warp.
Expansion warp.
Yeah, it means one step in the reference timeline aligns with multiple steps in the query timeline. Essentially, the query cell is lingering or stalled in that state.
Okay, so it's moving slower.
Right. And W is a many-to-one compression warp, which is the exact reverse.
So the query cell rushes through a state that took the reference cell several steps to complete.
You got it.
Okay, so those first three, M, V, W, are basically just variations of matches, right? They account for biological processes moving at different speeds.
Yes, they handle the stretching and compressing.
But the real breakthrough is the mismatches. I and D.
The indels.
Right. State I is an insertion. This is a complete mismatch indicating an unobserved state or a radically different gene expression popping up in the query cell.
Exactly.
And D is a deletion, where a state present in the reference is completely absent in the query.
Yes.
You know, it's highly analogous to a rigorous text editing process.
Oh, how so?
Well, if you're comparing a first draft of a novel to the final draft, you don't just pair sentences one-to-one.
Right, that wouldn't work at all.
Sometimes the author takes a single scene and expands it over 3 pages. That's your expansion warp, state V.
Okay, I like this.
Sometimes they condense a whole chapter into a simple paragraph. That's a compression warp, state W.
Makes sense.
But crucially, sometimes they write an entirely new chapter out of nowhere, an insertion, state I, or they delete a character entirely, a deletion, state D.
That is a perfect analogy.
G2G is basically the first tool giving single-cell biology the complete editorial alphabet.
And the ability to map all 5 of those editorial decisions mathematically completely changes how we interpret cellular behavior.
It's a huge paradigm shift.
It really is. Before we dive into the specific math powering those decisions, we should crystallize the shift happening here.
Let's recap it for the listener.
We are moving from older pseudo-time algorithms that falsely force biological alignments.
The Band-Aids.
Right, the Band-Aids. Moving away from that to a 5-state machine that explicitly captures exact matches, temporal warps, and most importantly, complete sequence indels.
Which should prompt another question for you as you analyze your own data, regardless of your field. Are you using tools that force comparisons because the algorithm just demands a continuous line?
It happens more than you'd think.
Because acknowledging the exact parameters of a gap, knowing exactly where things fail to match up, is often where the actual discovery lies.
And discovering those gaps mathematically requires a highly specific mechanism.
Right. How does GDG actually decide if 2 data points belong in one of those 5 states? It's not magic.
No, it relies on a concept called minimum message length, or MML inference.
MML inference. This is where the underlying mechanics get really fascinating.
Yes, because single-cell RNA sequencing produces incredibly noisy, sparse data.
It's messy. You can't just look at the simple mean average of a gene's expression to determine a match, can you?
You absolutely cannot. If you just look at the average, you miss the whole picture.
So what does G2G do instead?
When GDG compares a data point from the reference trajectory to the query trajectory, it evaluates the entire distribution of the gene expression across that cell cluster.
The whole spread.
Exactly. It models them as Gaussian distributions.
Okay, bell curves.
Right, bell curves. This means the algorithm is accounting for both the mean average and the variance, the natural spread and noise of the data.
That's crucial because biology is inherently noisy.
Exactly. And to decide if those 2 Gaussian distributions are a match or a mismatch, it calculates a strict information-theoretic cost.
Using minimum message length. Now, minimum message length is rooted in data compression and Shannon information theory, right? Where information is measured in units called nits.
Nits, yes. It's like bits, but based on natural logarithms.
So the algorithm is essentially running a cost-benefit analysis on the data.
Precisely. G2G poses 2 competing hypotheses for every single pair of data points.
Okay, hypothesis 1.
Hypothesis 1, they match, meaning they can be described by a single shared Gaussian distribution.
And hypothesis 2.
Hypothesis 2, they mismatch, meaning they must be described by 2 completely independent distributions. The algorithm then calculates the Shannon information, the message length in nits required to encode the data under both hypotheses.
Okay, let's use another analogy here.
Yeah.
It's like zipping files on your computer.
Oh, exactly like that.
If the reference cell and the query cell are doing the exact same biological task, their gene expression distributions will overlap beautifully.
They'll look almost identical.
Right. So encoding them jointly under hypothesis 1, zipping them together, will require far fewer nets. It's a highly efficient compression.
Yes.
The math looks at that and says, this is cheap to encode together, therefore it is a match.
Yeah.
State M.
And here's the genius part. If the diseased cell has a massive spike in expression while the healthy cell is dormant,
They look totally different.
right, forcing those 2 radically different data spreads into one joint Gaussian curve creates a massive, sloppy, artificially wide distribution.
Terrible bell curve.
Terrible. And the Shannon information required to describe that forced, unnatural curve skyrockets.
The mathematical bloat becomes too expensive.
Exactly.
So the algorithm calculates that encoding them independently costs significantly fewer nits than forcing them together.
The cost difference becomes undeniable.
So the algorithm confidently declares an insertion or deletion.
Yes. There are no arbitrary human thresholds here.
No scientists guessing.
None. It is a strict, undeniable mathematical cost based on the variance of the data itself.
That is incredibly elegant. Let's do a quick checkpoint on that before we look at the real-world results.
Sure.
So MML provides a strict information-theoretic cost to judge if data distributions truly match, preventing those sloppy, forced alignments.
That's the core takeaway.
So think about that, you listening. Think about the power of knowing exactly when a process goes wrong mathematically, rather than just knowing that the end result is broken. How would that change troubleshooting in your own field?
It changes everything. And the researchers actually put this mathematical rigor to the test.
Yeah, let's talk about the proof. They ran simulations, right?
Massive simulations. They generated 3,500 simulated trajectories, testing complex patterns of divergence, convergence, and perfect matching.
And how did G2G perform?
Across all scenarios, genus 2 genes achieved near-perfect accuracy, hitting between 98 and 100%.
Wow, 98 to 100%.
Yeah. And tragedy, the older thresholding method, by comparison, hovered around 66% for simple matches.
That's a huge drop.
And it plummeted to single-digit accuracy in complex divergent scenarios. It completely misclustered genes and hallucinated matches that weren't there.
That's devastating for the old models.
Yeah.
But simulations prove the math is sound in a vacuum. The real test is applying MML to messy real-world biology.
Absolutely.
And the paper details a fantastic case study involving dendritic cells.
Yes, the sentinels of our immune system.
Right. So they took these cells and stimulated them with two different triggers. One was PAM, which is a synthetic lipopeptide that mimics a gram-positive bacterial infection.
Okay.
And the other was LPS, which is a component of gram-negative bacteria.
Now, biologically, PAM and LPS hit different toll-like receptors on the dendritic cells.
So the immune system should react differently to them.
Exactly. They wanted to compare the timelines of these two distinct immune responses.
And what happened with the old methods?
When they ran the old DTW methods, the algorithm just blurred the timelines together. It suggested the cells reacted on roughly the exact same schedule.
But I assume G2G's MML cost calculations refused to smooth over that data.
You assume correctly. G2G explicitly flagged a massive insertion in the LPS timeline.
An insertion? What was driving it?
It was driven by a group of genes known as precocious expressors, specifically genes like IRF7 and STAT2.
IRF7 and STAT2. What do they do?
They are critical for antiviral and antibacterial defense. In response to LPS, the single-cell data showed these genes spiking immediately.
But in the PAM stimulation,
their expression lagged significantly.
Wow. So DTW treated that early spike in the LPS cells as, like, statistical noise to be smoothed over for the sake of a clean alignment.
Yes, it just erased a key biological difference.
But G2G drew a box around that exact pseudo-time block and declared a fundamental mismatch in the early immune response.
Exactly.
It's the difference between knowing a software program crashed at the very end versus finding the exact specific line of code where the bug originated.
That is exactly what it is.
Which perfectly sets up the clinical implications of the next case study, idiopathic pulmonary fibrosis, or IPF.
IPF is a really tough one. It's an incurable, progressive disease where the lung tissue becomes irreversibly scarred and fibrotic.
Is constant scarring.
Yes. The researchers wanted to map how cells differentiate in a healthy lung versus an IPF lung.
Okay, walk us through the healthy version first.
In healthy lung regeneration, a progenitor cell called an alveolar type 2 cell, or AT2, differentiates into an alveolar type 1 cell, or AT1.
AT2 to AT1.
Right. The AT1 cells are the thin, flat cells responsible for actual gas exchange in the alveoli, so you need them to breathe.
But in an IPF lung, that crucial AT2 to AP1 pipeline gets completely hijacked.
It does. The AT2 cell starts to differentiate, but veers off course. It transforms into what is called an aberrant basiloid cell, or ABC.
ABCs.
Yeah.
And those are the problem.
Big problem. These ABCs gather at the edges of the fibrotic scarring and actively drive the disease progression.
So the ultimate diagnostic question is, at what exact transcriptomic moment does the healthy trajectory permanently diverge into the diseased ABC trajectory?
Right, and to find out, G2G aligned the healthy AT2 to AT1 trajectory against the diseased AT2 to ABC trajectory.
And because MML doesn't force continuous alignments?
It was able to pinpoint the exact early and mid-stage sequence insidials where the timelines separated.
It found the exact split.
Yes. Yeah. And more importantly, because G2G operates at single gene resolution, it captured the sudden anomalous upregulation of a specific gene called CMK1D in the diseased cells.
CMK1D. The cost of forcing a joint distribution for CMK1D expression between the healthy and diseased trajectories must have been mathematically astronomical.
It was.
Forcing G2G to flag it as an insertion in the disease timeline.
But why is CAMK1D the smoking gun here?
Because CAMK1D is a known downstream target of TGF-beta1.
Yes, transforming growth factor beta-1. It is the master regulator of fibrosis. It is the primary signal telling the lung tissue to scar.
Oh, wow.
G2G didn't just show that the end-state cells were different. It pointed directly to the exact gene at the exact pseudo-time moment where the TGF-beta1 pathway hijacked the cell's development.
That is incredible. Let's synthesize this for a moment. We've seen how minimum message length uses Shannon information to calculate the exact cost of a biological match, totally rejecting those forced alignments.
Right.
We've seen it drastically outperform older algorithms by catching the precocious expression of IRF7 in dendritic cells.
Yes.
And now in IPF, it mathematically pinpointed the exact moment TGF-beta1 signaling diverged a healthy lung cell into a scar-driving basaloid cell.
It's a level of precision we haven't had before.
For anyone dealing with complex system failures, the takeaway here is the power of high-resolution divergence mapping. If you can isolate the specific point of failure with mathematical certainty, rather than just observing a broken end state, you aren't just reacting to a problem anymore. You possess the exact blueprint required to troubleshoot and intercept it.
And intercepting the problem at the blueprint level is basically the holy grail of regenerative medicine.
Which brings up a huge point. Identifying a diseased cell's divergence point is incredible for diagnostics, but can this math actually help us build better cells from scratch?
Yes, the paper actually explores this by comparing cells grown in a lab to cells naturally grown in the human body.
The in vitro versus in vivo case study.
Exactly. The researchers applied G2G to the developmental trajectory of T cells.
T cells, the foot soldiers of our adaptive immune system.
Right. Naturally, they develop in the thymus. That's the in vivo environment.
Okay.
But scientists are heavily invested in engineering these cells in a petri dish, in vitro, for targeted immunotherapies, like fighting specific cancers.
But lab-grown cells are notoriously stubborn, right?
Oh, incredibly stubborn. They frequently stall out in development and rarely match the natural versions perfectly.
And without knowing exactly what is missing from the petri dish cocktail, scientists are largely just relying on trial and error.
Lots of trial and error. But when the researchers aligned the pseudotime trajectory of the lab-grown T cells against the natural T cells, G2G revealed something fascinating.
What did it find?
It showed that the in vitro cells do successfully match an immature intermediate state of the natural cell.
Oh, so the early development was actually accurate.
Yes. But critically, G2G generated a massive deletion state at a specific late-stage developmental hurdle.
A deletion, meaning something was entirely missing.
Exactly. The algorithm proved that the lab-grown cells completely lacked the expression of a specific set of genes associated with TNF signaling.
TNF, meaning tumor necrosis factor.
Yes. TNF is a critical signaling pathway required for immune cell maturation and survival.
So the lab-grown cells were stalling out, failing to become fully functional T cells because they weren't receiving the necessary biological signals to activate the TNF pathway at that specific point in their development.
You nailed it.
So G2G is essentially serving as the ultimate quality control inspector for regenerative medicine.
That's a great way to put it.
If we want to engineer perfect lab-grown cells to cure cancer or replace damaged organs, guessing what is missing from our biological recipe wastes years of research.
Decades, sometimes.
But G2G provides the precise, missing transcriptomic ingredients to optimize those in vitro culture conditions. It mathematically tells scientists, your recipe is perfect until pseudotime step 40. Right here, you must introduce TNF signaling.
It accelerates the entire field from observational trial and error to precision engineering.
This is wild to think about.
By unifying dynamic time warping with biological gap modeling via that 5-state machine, researchers are no longer bound by the flawed assumption that every cell state must have a parallel.
We finally moved past the era of smearing data together to force a mathematically convenient fit.
Exactly.
By calculating the Shannon information cost of variance, the Junis 2 genes framework respects the biological gaps. It highlights the divergence with single-gene resolution.
And in doing so, it allows us to spot the precise transcriptomic origins of disease and the exact missing ingredients in lab-grown therapies.
Which leaves us with a truly profound possibility to consider as we wrap up this deep dive.
Yeah, where does this lead us?
We've seen how this framework mathematically pinpointed the exact moment a healthy cellular timeline veered off course in idiopathic pulmonary fibrosis, catching the CAM-K1D activation before the cell fully mutated.
Right.
If we scale this computational power, could we eventually use this 5-state machine to map the exact transcriptomic divergence point of every known incurable disease?
The potential is definitely there.
Imagine a medical future where we possess the mathematical map to intercept illnesses at the microscopic single-cell level, correcting the biological timeline before a single physical symptom ever has the chance to appear.
It's a completely new frontier for medicine.
It really is. So, to you listening, Keep questioning the continuous lines in your own data, embrace the mathematical gaps, and thank you for joining us on this deep dive.